Yeah, Vinita, we are good to go.
Okay. Hello, everyone. Good to see you all. So, first, we will go ahead. Before that, we would like to share your internship structure. Let me do this one. Okay, so your internship is of 5 to 6 weeks. So, this was our first week that we covered all the terminology and basic concept of biology and bioinformatics last week. And the second and third week would be all about bioinformatics, that we will download some of the sequences and align those sequences using some of the top softwares. And for that week, you will choose your species that according to the project that we design for you guys. So you will download the sequences for your own project and you will perform the same steps, the alignment. And then week 4 would be all about evolution. So again, we will use some of the bioinformatics based platform to reconstruct the evolutionary history of species using the DNA or protein sequence database. And then your week 5 would be all about R programming language. You will learn how to write a simple code in R and how to analyze the data. For that, I will provide you some of the data that from my research, and we will see in using R how we can analyze that data like in graphical form and how to interpret that data. So this was all like the 5 week sort of outline of your internship, that what we are going to cover. And next is in the last session, I talked about bioinformatics platform and I highlighted only the tools that we will use in this internship. For genomics, we are going to use these platform, NCBI, UCSC Genome Browser, and Ensemble. So whatever I wrote below the heading is all the tools that we are going to use in our internship. So let me go back. So first, I wanted to show you the first platform of bioinformatics, which is NCBI. It's a National Center for Biotechnology Information. So this is a platform where all the researchers worldwide, they submit their data. And when we are saying data, that means they submit the sequences of the genomes. Someone is working on bats, someone are working on mammals, someone working on fish. So on what organism or some plants, if they are working on, in what taxon they are working on, and if they retrieve the DNA from that species and they perform the genome sequencing, and they always submit their data onto the NCBI. On NCBI, there are a lot of further sub databases on this platform. So you can see the first is assembly. So this is always for the genomes. If you wanted to search for any genome, for example, if I will click on assembly and I wanted to see, so this is a species name from the Darwinian finch. This is a bird. So you can see when I search for this species, you will see that the assembly, there is a one hyperlink and there is a uh, combination of alphabet and numbers. So this number is, we say, accession number. That means this is a unique identifier for any species, what kind of genome it have. So you can see that there are further accession number that on GenBank. This is the accession number for this species. This is the scientific name, and in brackets sometimes the common name of that species. If the sequence belongs to male or female, and if annotation, that means for this species there is information available for each gene, what gene it is, where it is present in the genome. And then next is level, that means there are two level of the genome. One is at the chromosomal level. For each species there are certain number of the chromosome. For example, for Antarctic fishes there are 46 chromosome total. So that means this level is not chromosomal, this is scaffold level. That means DNA is present in small scaffolds, like short of packets, instead of the chromosomes. And when this genome got submitted on the NCBI, that means the release date, it's in 2012. and then further the whole genome sequencing accession. And further, if you wanted to know the detail of this one, I think it's just saying that this is the whole genome shotgun project and it contained no sequence data, but you can find all the information like who submitted this genome, what lab submitted this one and when, and the all details, what assembly platforms someone used to sequence this data. So this is how we can use the NCBI platform to look for data. So this is what I showed you for the genome, that how we can search for the species for the sequencing data. Next is there are another databases which are genes, then again genomes, then nucleotide, protein, and there are so many lots of other databases in the NCBI. Next, I wanted to show you UCSC genome browser. This is again a platform where the genome sequences are available. But the difference between NCBI and UCSC is that on NCBI, the whole genomes are available. It's not necessary that they have to be annotated. But on UCSC genome browser, it's very compulsory that if you have a genome and you wanted to submit that genome, you need to provide the annotation. You need to provide every gene's information for that genome. So that's why there are only limited genomes which are well annotated. For example, you can see human, mouse, rat, and there are other. If you will scroll this phylogenetic tree, you will see for the cow, pig, and all the mammals you will see. So this is another platform. And apart from this, there is one genome browser, Ensemble. It will take time, I guess. Yeah. So this is another genome browser that have information of all the vertebrate genomes. And this platform is also similar to the previous one, but again, the way how they annotate their genomes and available for the public are slightly different. So now we will see some of the example how NCBI, UCSC, and ensembles are different. So for example, on NCBI, if I will go to maybe just NCBI genome, and then here genome, and then I wanted to see some of the gene. So if I will copy this name of species, and if I will show to on Google, you can see that this is a fish, right? And now I wanted to search some of the gene. So there are three options. Either you can search by gene name or by gene identifier. That means if you know that there are certain accession or identifier for that gene, or if you have a list of gene that you wanted to know what kind of these genes are, so you can provide that list as well. So now let's search with LC582. So this is a gene that I search for this species, and you can see that for this there is a number, right? And this number is a unique identifier for only this gene, which is SLC5A2 gene, solute carrier family 5 member 2, and this is the species information. And what this gene is, this is protein coding gene, and how many transcripts are there, if is there any orthologs, and genomic coordinate. So this is the chromosome number, like this is the chromosome accession number, and on this chromosome from this position to this position this gene is present, right? And this is how we search, this is input. And if you will further click on this gene ID, you can see on left hand side you came to this gene tab which I showed you previously, that from all databases, if you will just click on the gene, you will see that all the information available for this gene, that this gene is also present in human and zebrafish. So what are ortholog? For that I wanted to show slide. Okay, so orthologs, let me hide this panel. Orthologs, that means there is a gene that shared a common ancestor. That means both the species, which is P. passifus and C. elegans, these are two different species, and they have this gene DAF16. This DAF16 gene was present at the common ancestor of all these, of both the species. So that's why this is ortholog. And what is paralog? Paralogs, that means there was a gene dash 1 which was present in the common ancestor, and then at this point, which is the branching point, from this point that species splitted into two different species, and this event is known as speciation event. That means after this speciation, this dash 1 go to Pacific, but before it will go to C. elegans, it further duplicated. That means it make its own copy, and you can see that there is dash 2. So that means now this dash 2 is paralog with the Pacific, because this is only species specific. It only duplicated before this species form, and it's only present in the C. elegans. And at the sequence level, so this is a phylogeny. Phylogeny, that means this is a graphical way to represent the evolutionary relationship of any species. In this case, you can see this dash 2 is actually present outside of this phylogeny. That means this dash 1 was present here. So this was the main point from where all these four branches came out. That means this was the common ancestor where this dash 1 was present, and then from here it went to two species, this specific species and then C. elegans. You can see this is the branching point, it went there, and only in the C. elegans this dash 2 is present. So that is how this is evolutionary related, that this is how we will read the phylogeny to predict how they are related to each other during the evolutionary time. And based on sequence as well, you can see that the first sequence is dash 2, and you can see that this is the uh, box which is like shaded, and there are no star. So wherever the star is, you can see the amino acid is same. That means these sites are highly conserved, but only this certain sequences which do not have star above them, that means these are the changes which are different in every species, and only because of the dash 2, because it is very different from the dash 1. Okay. So So this is how you will look for any genes and their description, and where these genes exactly present in other species. So in orthologs, if you will click on all, you will able to see in how many other vertebrates this solute carrier gene is present. So you will see a slightly description of this gene, where this gene is expressed in. It's expressed in the kidney, and if mutation happen in this gene, it can lead to renal glycosuria. And on the left hand side, there is a panel that you can see that there is jaw vertebrates, and under that there are birds. In birds, there are songbirds. Only for two species this gene is present, turtles and so on. Like there are a lot of species. You can see that for almost 496 vertebrate, this gene is present on NCBI. So you can select your species if that have this gene. Same gene, let me search the species. We will go back. And this is our genome. We'll search here. So this is our genome of this Maclovinus fish. And if we will search SLC582. Okay, so this is how it the annotation looks on UCSC genome browser. So can anyone tell me that what are these lines, like the dark blue lines, and there is a sort of empty line? Let me make it bigger. Anyone wanted to try?
Maybe the dark lines are where the expressive genes are present, the coding regions.
These? And what
Yeah,
about this, the intermediary line?
the non-coding regions, like
Yes.
the introns probably.
Yeah. So that means whatever the the thick boxes are, these are the exons. That means these are the coding sequences that are ultimately going to make protein. And whatever the connecting line between these boxes are the intron. So on NCBI, we just saw that you can see that on NCBI, it gives us information that this gene is present on chromosome 18, and you can see this SLC52 just represented by a arrow, right? And it also showed some of the flanking genes, that means the neighboring genes of this gene. But on UCSC genome browser, you can see that every exon's information, we can see how many exons are there. And if you will just roll over your mouse, you can see that this gene have total 15 exons, right? And if you will click on this, you will further see that this gene and NCBI gene ID is this one, the transcript accession is this one. And there are three links available for this gene on UCSC genome browser. First, let's open this one. Okay. So there are three options. Let me show you again. There are three options for every gene. First is genomic sequence. Genomic sequence, that means it would be about whole gene. Whole gene can include 5' UTR, all the exons, intron, 3' UTR, or poly A tail, right? So this is whole gene. Second is predicted mRNA. That means this is only a portion of flanking regions of the 5' and 3'. That could be only the portion of UTRs. And on UCSC, there is a format that if there are UTRs present for that gene, they would be in small letter. So in this case, you can see this is 5' UTR of this gene at the sequence level. And you can see that this is the first ATG, which is the start codon of any protein in eukaryotes. So this is the starting of any gene, protein coding gene or coding sequence. So this is ATG. And can anyone try what could be the stop codon?
UAG.
Yeah. So in you can see at the DNA level, it's TAG, right? It could be UAG, UGA, or UUA. So like that, out of three, anyone could be acting as the stop codon. So at the DNA level, you can see this is TAG. So when we use online platform, rather than uracil, like for RNA, it's always thymidine because uracil is always present at the in the cells. But all the work that we are doing, it's virtual. So that's why to make it convenient for every researchers, we don't have U in the sequences at the RNA because its U is only present at the cell level in the in vivo conditions, but at virtual platforms, it's always T. So it will be easier to see the uh, at the DNA level, like sequence level. So that is how you can see that this is the stop codon, and after that, this is 3' UTR. So this is how the mRNA look like at the sequence level for every gene. Next is, let me open translated protein from the genomic sequence. So these amino acids are only for the exons only. So you can see that starting is the methionine and it's coded by the ATG triplet codon. So this is how the starting amino acid in any protein is methionine. And you can see that length of this gene protein sequence is 653. So that is how these you can explore the genes over the different platforms. And on NCBI, you can see for this gene, there is previous exon, like before the exon, there is this rectangular box. So these are the UTRs. Some of them, there is no UTR. For example, for this gene, there is no 3' UTR, right? There is just exon and no UTR. So it's just depends on different genes, how well annotated they are. And you can see the descriptions for every gene in the UCSC genome browser. Second thing, there are different tools for example, if I sequenced a genome and I wanted to know where the genes and what are the genes in my genome, there are different tools to annotate those genomes. So what tools or platforms you use to annotate the genomes, it depends how it will annotate your genome. So that's why every platforms are different for every, like the gene is still same, but the way of annotation is slightly different. On ensemble, this species is not available. Let me show you. For example, we can search here. So you can see that there is no species, right? So maybe we can look for another species. Okay. Oh, this is another fish that present on the ensemble. And here there are different kind of statistics present. For example, download the DNA sequence, then download the FASTA file for genes, cDNA, non-coding RNAs, and there are other gene files. That means for the whole species, all the genes which are present in this species, you can download all the files. In this one, there is category gene. Okay. For this one, we need ensemble ID. To see if it will. Okay. So sometimes when we search with the gene name, sometimes it won't gives us the information because, for example, for NCBI, the gene ID of this gene is something like this number, just 13880106. On ensemble, you can see this number is XM, or for protein, it's XP, and there is certain number. On ensemble, this is the criteria of any genes. That means first would be ENS, that means it's representing the database ensemble. And then this CG, that means this Gobia. So this is Cota Percogobia, species name, genus name. And then OG, that means this is the gene ID. If there is T, then it's transcript ID. And then there is a certain unique identifier for that gene. Okay, on ensemble, you can see this is the same gene again, which is in different fish. And for this gene, you can see there is extra information available that this gene has 9 transcript. Now, what is transcript? So anyone can tell me like why we have introns in the genes. If they are not making protein, why they are there? Is there any role of introns?
No, introns are non-essential.
So why, I mean, why our bodies spending energy to maintain them, why it's not possible to just remove from the sequence so that we have very less sequence, only protein coding sequences, and it would be helpful to spend less energy.
Maybe because introns increase the length of the DNA. So because after each transcription cycle, the length decreases. So then maybe to act as a buffer.
Good try. So okay, let me explain you why introns, we have introns in our genes and they are not like translatable. Sometimes in cells, depends on environment and depends on how much protein they needs. Sometimes it needs shuffling, that means it only require a combination of exons. For example, if there are 15 exons, that means there are 14 introns, right? Sometimes maybe in this particular species, in this fish, it only require a form of protein that is it's sufficient for this species to perform the function, and those could be maybe exon 5 to exon maybe 7, like 1 to 7, or maybe 1 to maybe then 7, 8, 9, 10, and further on. So that is how intron provides the diversity. That's why we have intron. That means we can have a diverse range of the proteins from the same gene, and that we call the isoform. Isoforms are the different forms of a gene, and different forms of a gene could be only possible with the introns. How many introns are there? That means you can create a lot of diversity. That means there would be a lot of combinations you can make with your exons, and there would be slightly differences in the protein sequence level. And now let's assume that, for example, there are all 50 exons are expressing the protein, and this protein is almost 700 length. And sometimes it's not very essential for that species to make 700 amino acid. That means maybe rather than spending energy on making 700 amino acid long protein, it will only spend some of the energy and only able to make a combination of exon 1, exon maybe 3, and then exon 8 and 11, 12, 13, 14, like that. And the protein sequence length would be shorter, like it's just maybe 400 length, but it will perform the same functions what the 700 length was performing. So that's why, because of the introns, cells have this ability to make the diverse form of the same protein. Okay. On Yes, someone asked question.
Yes, Miss. I had a question that, Miss, if I think in alternative splicing we remove the introns, right? So, to,
Yes.
to only have exons. So then, Miss, what is the point if we're removing it anyway? Then
No, because this is for the combinations. For example, if there are no introns, then you cannot make the choice.
you cannot distinguish. That's why.
Yes, that's why we have the introns, so that if cell wanted to have a combination, maybe exon 1 to 5 and then maybe 9 to 10, right? And if there are introns available and they have those splice sites, those signatory sequences are there. So spliceosomes will recognize only those splice sites that cell needs to cut it out, that I only need to cut out these exons. So it will only break those and they will make the protein. That means it will further translate.
Understood, Miss. Thank you.
So on ensemble for on ensemble, this is the information available for this is the additional information available for this gene. You can see that this gene has 9 transcript, and if you will see at the protein level, right, there is this term ensemble canonical. Canonical, that means this is the most classic form, which is highly conserved in all the vertebrates, and you can see the length of this one is 691. Then there is another isoform. You can see the transcript ID. So isoforms are also known as transcripts, and you can see on the shortest one is the 551, that there is a less number of protein in this sequence. So that is how transcripts are look like at the sequence level. For example, let me open this one, this one, and there is option here sequence. In sequence, there are exons, there is cDNA, and there is protein. cDNA is equivalent to the mRNA. So let's go to protein. And you can see that this is the protein sequence. But on ensemble, you can see that there is uh, an exon, another exon, and residue overlap splice sites. So there are 3 combinations of exons available on ensemble. So whatever the black letter, that means these are the original exons were there, maybe exon 1, 2, 3, 5. After that, what could be the possible, the combinations? And for this specific transcript, we don't know what exon combinations are there. So based on that, all the blue color, that means these are the alternate forms. These are the combinations of maybe exon 3 and 5. So these sequence highlighted in the different color based on how this transcript is formed in that species. There is another cDNA. On ensemble, cDNA look like this. So whatever the white color and whatever the yellow color is two different meaning. First is alternating codon. That means the original codon were there, and the highlighted one, that means this is for the exon, and the highlighted one is for the another exon, the blue color. So you can see the ATG. This is the initial codon, and then you know that the genetic code is degenerate and it's always in the triplet form, and that is how this is how to read the frame. So first is ATG, then GAG, then AAT. So this is like the continuous way, but it annotated them as an alternating codon. For example, this is ATG, so protein could start from ATG, then it could include the GAG or maybe AAT. So either during the isoform, either it can have E or N. It depends on cell and species requirement, or it can have the canonical one, that E and N. So this is how protein, the first is like the mRNA sequence, and then at the lower end for every codon, it showed the protein sequences. And these are the exons information. So on ensemble, there is a slight annotation variation for the 5' UTR. So you can see that these are the green letters. And then there is information available for introns as well. In this case, there is a intron which is in gray color. And then again, intron 2 to 3, it's from starting from the total length is 451, and you can see the start and end of every exon and intron. So that means on what chromosome and from where to where the exon and introns are, and what are the sequences for these. So on ensemble, you can get these information as well. Apart from that, let me show gene. Okay. So now sometimes you are looking for a gene and you wanted to know if your gene is related to some of the most critical function in vertebrates. In this case, on ensemble, there is one tab available, that phenotype. That means phenotype for this gene. And you can see that phenotype or disease and trait. Trait is again phenotype, and sources, that means in what species. What are the sources? That means the links available for different platforms, for the human, for the mice, and for the Norway red mice. And then what is this gene's accession number for that? Like for the human, the accession number is different. For mouse, it's like different. And you can see that human, it can leads to this disease and so on. Like there are a lot of disease that involve in mouse, it's lipid deposition and so on. So you can also get some extra information for the genes. And from this, you can see that there are 197 orthologs and 25 paralogs. That means these 25 orthologs are before speciation event happened. They are just species specific in those species. And from NCBI, okay, so this was all about how to search for the species if the genome is available. Second thing, if genome is available, look for the annotation. And how to look for annotation? Through the NCBI genome, it will tell you whether genome is annotated or not. And if annotated, then look for the genes. You can maybe type the gene name and it will pop up the gene just description, and you can click on the links and you will go to that page of the gene description. And so these are the three main platforms that people are using for their research. Okay, now next thing is that how to download the sequence of any gene. So on NCBI, there is option download dataset. In that, either you can download the gene sequence, transcript sequence, or protein sequence. Now let's see, we can download all three and download. Apart from this download tab, there are options, two options. One is genomic. That means if you will click on the FASTA, it will take you to the another page. And there is one option that if you wanted to download only mRNA, that means the accession number start with XM. And if you wanted to download the protein, then it would be XP. So if you will see FASTA. Okay. In bioinformatics, whenever you will talk about any genomic sequence either protein or DNA, that sequence will always go into file with .fasta extension. So this is how this sequence look like if you will scroll down, because this is for the whole gene sequence for this SLC52. And there is another database, UniProt. So on UniProt also, you can extra information, you can see annotations, the highlighted one, that where this gene involved in. So you can see that under all molecular function, this gene belongs to the transporter activity. That means this gene have function, something to do with the transport. And then all biological process. And then there is, let's see. Interpro. So this is a database, Interpro, which is based on the sequences. It will predict what your protein belongs, like what family your protein belongs to or your gene belongs to. So it can give you maybe a just brief description of that gene. And you can see the family membership is the sodium solute symporter. Symporters, that means inside cell, if something is going in, it will take something with that. That means if sodium is going inside the cell, it will take other solute with that. So that solute could be glucose or something else. But in this case, it's glucose because this is sodium glucose symporter and they are going together inside or outside the cell. So this is Interpro. Okay. So we just downloaded sequence of a gene, how it look like. So it will always give you in .zip folder and there are NCBI dataset. And in NCBI dataset, there is a data folder. In that there are, you can see, either you will see .fasta, .faa, .fna, .fs. So all these extensions, that means that file have the sequence information. So if you will see the gene.fna and open it in any of your notepad editor. So how to read this file? So that means the greater than symbol. So this is the signature of this FASTA extension. That means this is the starting. That means if you wanted to save some sequences in FASTA format, first you need to start with the greater than symbol and then there would be a information. So this we called header. So this is header of any file where it will have information of gene ID, species information, sometimes if the chromosome information is available. So you can see that this is chromosome accession, this is the genomic range, this is the gene name, species name, and then chromosome information. So this is how to read any .fasta of sequence file, .fna file. For protein as well, it's same, that you can see the accession number is XP, which is P for protein, and the gene, and then species information. Okay, so this was all about how to search for sequences on different platforms if your species are available and how to download. In this case on UCSC, you just go there and maybe just predict mRNA. And let's say if you wanted to save all, Control C, just select all and then open any of your notepad editor and then save and save with maybe gene.fasta. Remove this .txt. So this is how you will save any sequence file in your system. Now in the download, you can see that gene.fastAs have the sequence. Okay, next part is what will be your project. So let me share my screen. Okay, so your project would be all about the finches, mainly the songbirds that have 2 or 3 species from the Darwinian finches. So if you will look at this figure, anyone wanted to try that what they have different in them?
Their beaks and their eyes.
Okay. And what do you think that if something is very like distinguishable, what could be the factor that drive that difference? If you are
Their
saying that,
food sources.
Evolution, natural selection.
okay, anyone also was saying something?
I think it may be their food sources.
Mm-hmm. So yeah, that means if we are mainly talking about beaks, which are like clearly visible in this figure, that every bird have different beak, and what are the driving factor for that difference is only the food. That means their diet. So this is a map on the right hand side, that Galapagos Island. So when Darwin like visited this island, and he went into different islands, and he observed that there are different birds that have these beak variations. And then he realized that these beak variations are mainly because on each island they had different diet. And based on that, they have this diversity. So for this internship, your project would be DNA to diet. That means you will look into some of the genes. That means these would be the molecular signatures and which are responsible for the beak diversity in finches. So for the time being, because there are lots of availability of the songbirds, not for all the Darwinian finches. So if I will tell you how to look for genes and what could be the genes, so you can able to download the sequences. So in this project, we will consider some of the genes which are very known, that they are involved in the different beak shapes and sizes. That means beak morphology. And we will use those sequences, those genes, and we will download sequences for different birds. And then we will align those sequences and see where are the variations are at the sequence level, because the gene is still same, and in every bird this gene is responsible for the beak morphology, and we will see how based on variations those beaks morphology are diverse among the songbirds. Anyone wanted to ask question or clarify this thing? Okay. So first thing is that how do we know that what genes are involved in peak morphology? So first thing is to just search in Google, but not simple Google. It would be Google Scholar, because every research information is available on Research Scholar through the papers. So now we will search beak shape and size morphology. Okay. So now you can see there are so many paper came out, right? And you can see that 2 developmental module established this 3D beak variation. This is from 2011. Let me see if this is the same paper. No. Let me pull out paper that I have. So I will share the paper title in the chat box now, so you can look into those. Okay, so if you will search this title, you will get a paper. And then there is another paper. Okay, so I just shared two title of two papers. You can just copy them and paste into your Google. So first paper that I shared, you can see, hide it. The title itself saying that a beak size locus in Darwinian finches facilitated character displacement during drought. That means this is related to beak size, and it's more about Darwinian finches. And in that paper, they explain that how drought, that means because of environment, there is a change in the beak, that means the trait and morphology. So in any paper, how you will look for the information. So for every paper, there is one section which is available for each paper is abstract. So abstract is always, this is a very brief about the finding of that paper. So the first 1 and 2 line would be the background information. Then next, what they used, what approach they used to answer some of the questions that they hypothesize. And then at the maybe in between, you will find we discovered or we found like through this study. And you can see in this paper, they are more talking about, they used a genomic analysis to investigate the genetic basis of character displacement in Darwinian finches. And they are talking about the medium ground finches, which diverged from their competitor, large ground finches, during a severe drought. That means how medium ground finches diverged from their large ground finches during the drought conditions. And then they discovered genomic region containing HMGA2 gene that varies systematically among Darwinian finches with different beak sizes. That means by reading this line, we able to understand that this is the gene and this is the trait that involves in. That means now we found a gene which involves in beak size. And other trait is beak morphology. That means the similar way, if you will search maybe Darwinian finches and songbirds, and then keywords use beak morphology and genes. So you will, and beak shapes. So you will see some of the paper, and through the abstract, you can able to understand what this paper would have. So this is the first gene that we found, that this is related to the beak size. And further on, if you wanted to read more about, there would be a discussion section. Okay, in this paper, I think this is a newsletter. It's a like not a data paper, it's a report. It's a scientific report. That means it's a brief about their discovery. Second is the paper. Yeah, so you can see that this is a article. That means this is paper that will have the data information as well. So in this paper, you can see from the first line, beak shape in Darwinian ground finches. That means this paper would have information about beak shape and Darwinian finches. And then I think in introduction. Okay. So this is the section from the introduction that I highlighted here, that you can see from these studies several craniofacial genes. That means we are talking about birds. Now we are using the term craniofacial. That means it's more about their beak sizes. Genes have been identified which affect beak shape development in finches, and you can see the gene names. That means for shape, we got BMP4, calmodulin, ALX1, DLK1, HMGA2. So these are beak shape development genes. Okay, so in this section, they did not just break down between the size and shape. They just collect or gather the information of all the genes which involved in the beak morphology. And if let's search for this ALX1. Okay, I think in the intro itself, you will see that there is a section where they are saying 3 of 8 a prior candidate gene for beak shape were implicated in our analysis. That means now we know that these are the beak shapes genes: calmodulin, ALX1, DLK1. So that is how I won't ask you to read so many papers, but at least 3 and 4 papers you will see, and you're able to find maybe one candidate that you are interested. Second thing, it's up to you if you wanted to involve both traits in your project, or only you wanted to investigate beak shape or size. So it's up to you. Okay, so this was these 2 papers that you have. You can read and you can get the genes information. So now let's say how to look for birds, right? How to look that what birds or songbirds we should consider. So first maybe okay, so this is the first way that either you can choose birds based on their beak shapes and beak sizes. For example, from Google, you can look into the pictures. For example, in this case, that you can see the beak shapes are different, like it's more pointed. This is thicker beak, and um, I'm just looking for maybe any figure that. Okay, so basically, task would be for you to look for birds that have different peak shapes and sizes, and write down maybe 15 to 20 bird species, right? So you know now you have certain birds that you wanted to see that they have different beak shapes and sizes. And if you know that there are different beak sizes, then you will download the gene sequence for the HMGA2, because that gene is for the beak size. And if you are more interested in the beak shapes morphology or diversity in the songbirds, then you will download certain birds name, maybe 20 birds, and you will see that how shapes are vary in those birds, and then you will download the beak shapes gene. Then how to like there is one this is a website of time tree. So this is a phylogenetic based pipeline. So what this platform basically do, whatever the genomes are available on NCBI, based on those genomes, it will construct the evolutionary history of any species. So if I will go to, let's say, NCBI gene, and then ALX1, maybe just click on Homo sapiens. Okay, the first one. And now you can see that there is the information of orthologs. We will click on orthologs. Now in orthologs, there is a term taxonomy that you can search your names. song birds. There is the order. Darwinian finches. Okay. So sometimes common name won't work on NCBI. So you can see that now it's pop up. I don't know why. So it's Passeriform order. So you can see in this there are 55 songbirds information available, right? And the genes, you can see that gene is there and the amino acid length of this gene. And there are other birds as well. If you will like go to birds, and there are other birds, this serimas, falcon, songbirds, parrot, hornbill, mousebird, kingfisher. There are a lot of varieties of the birds available here. So it's you are not confined to only songbirds. It depends on you what your interests are. But the concept is same, that we will be investigating their beak morphology, either size or shape. So this is how we know that ALX gene information is available for 50 songbirds. How to download the sequences? For example, let me see the Geospisa, this medium ground finch. This is available. And if I'll go to download, okay, in download there are two options. First is ref sequence transcript. Transcript would be in the DNA format, and there is ref-seq protein, which is in FASTA file. And remember, we are looking for the traits. That means we are studying the phenotype, and for that we need the protein sequence. Remember, gene will make the RNA, RNA will make protein, and finally protein is the only that is going to responsible for the traits. That means it will depends on all how protein is going to fold. It's it's responsible for the traits in those species. So you will download the protein sequence of your gene of interest, and then after that there are two options: one sequence per gene, all sequence per gene. But for this project, you will download one sequence per gene. What is present in all sequence? So those are the isoforms within the same species for that gene. So we do not want it to include the isoform for this one. So we excuse me. So we wanted to see only the one sequence per gene. That means the canonical one, the longest isoform. And just download. And then, let me see. I think there is one, this species is also available. No. Lattice. Okay, on NCBI, you can also search, for example, if you will go to assembly, and if you will type sometimes family name, you will be able to see that for this family, how many genomes are available for different species. So for example, for the Darwinian finches, the family name is Theropidae, right? And I wanted to know for how many species the annotations are available. So, okay, so annotations are only available for two species, which is medium ground finch and this Parvulus one. So let's see the ALX one for Parvulus. No. Okay. So let me take only songbirds. What this bird is. Okay. Okay, so how to decide, like how to take birds? So for example, from this data, where is NCV? Okay, from this we know that there are 55 songbirds. So just click on all and you will download a CSV file because we wanted to download all the species name. So just go to CSV file and then it will give you downloads. This ALX ortholog CSV. Click in, maybe you can open in your Excel, and from here there is a column scientific name. That means these are the species scientific name. For these species, ALX1 gene is available. And just copy all these name and open your notepad and just save all these name. Make a file, maybe songbirds species.txt and enter. So why we made this file? Because in the time tree, we wanted to see how these songbirds are evolutionary related. That means based on phylogeny, first we can see if there are similarity among the birds, then we can skip those, and how to take the combinations. Maybe someone are further in the tree and they are more dissimilar. Some species are sister species, they are very similar. So how to choose the species? So in this one, you can go to your species name list. So where is songbirds? Enter, upload. Okay, so what time tree gives us? So this is based on how many million years ago these species separated from their common ancestor. So this is the time scale. You can see the time is million years ago, 0 to 55. That means all these songbird had shared a common ancestor 55 million years ago. At every node, if you will take your mouse there, you will see, click after you will click this, you will see that estimated time of this clade. That means these vidua the species genus, these are arrived from their common ancestor around 5 something million years ago. Okay, after that there is our Geospiza fortis. That means this is the Darwinian finch. This is how it shared the common ancestor at this branch. That means somehow this whole group, all the species that are in this group, they somehow very similar at the genome level as compared to this group, which is Montacilla alba and all these species. So what you will do, either based on beak morphology, you can choose the bird names, or either based on phylogeny. That means if you will compare this sequence, Fasciata and the Darwinian finch, let's see what it is. Okay. And then this one. Okay. So did anyone see any differences?
The length of the beak, the pointed part of the beak, like the crow's one is more broad, but the rented is smaller and thinner.
That means both the size and shapes are very different, right? You can see in the crow, the size is very long of the beak. And shapes are different. Like this shape is like more pointed and slightly blunted. But overall, you can see that that there is a huge difference in their beak morphology, right? So how we found that? From the phylogeny, that this species and this species, they are not evolutionary related. They are far diverse from each other. So that is how either you can choose from this phylogeny, that you can choose species. I won't recommend you to select all the species that belongs to the crow family. because then you won't be able to see the changes in the DNA level. So what I am saying now, let's see at the sequence level. For example, let's see what this is. Oops. Okay, this is Merina crow and this is New Caledonian crow. That means both are crow, right? Let's go to species. We just type the name. Okay, select. And then the second crow, and we would paste this one. Oops. First, I need to download this. Protein sequence, one sequence per gene, download, and then for this one, select this, download, one sequence per gene, protein. Okay, now at sequence level, how to see the changes? So for that, you need to install a tool, Mega software. Let me copy this link. So use this link, and on what system you are working, either on Windows, Mac, just go to this link and you will able to see that by default it will change according to your system. If you are on Mac, then it will automatically download for the Mac, and if for Windows, just click on the download and in your system install this software as you install the other apps in your system. Just double click on that and let me know if you finish. After installing this, you will able to see in your apps this like Mega.
I've installed it.
Yeah. Okay.
Miss, it's asking for questions that
Mhm.
whether you're from university and what kind of a profession do you have and all that.
Sorry.
It's
I'm
asking
a
for, huh?
no, I think it's free. So I think it's just a form. It won't ask like much detail. You can just maybe just ask for student information. So yeah, you just need to accept, because this is like someone made this software, so they just wanted to make a maybe catalog like how many people are using this software. So you can select India, student, and maybe your school name or something, and just download. It won't give you any problem. Yeah, you just select a student and maybe just, don't know, university and college. Yeah, it will download. No need to provide maybe the name of school as well.
Yeah, Miss, it's downloading.
Okay.
Done downloading.
Okay, so everyone downloaded, right? Okay, I am assuming that everyone have this this software in their system. So what this is a tool to align the sequences, and the MEGA is like this molecular evolutionary genetic analysis, the name of this tool. And this is basically used by all the evolutionary biologists if they are working with the sequences. And based on sequences, they wanted to predict the evolutionary relationship of any species. For that, we always first we perform the alignments. That means we wanted to make the sequences in such a way that we will able to see some of the variations at sequence level. What are these? Let's see. So in this one, if we will go to the first button, which is saying align. So go to align, edit and build alignment, the first option. And then it will ask you three things, whether you wanted to create, whether you wanted to open or retrieve. In this case, because we just downloaded the sequence, so we will say that we wanted to retrieve sequences from a file and OK. In this case, it will ask for the path. So here in my system, there are this 3.fasta is the file. And you can see from this line, you can scroll it further like this way. So you will be able to see the whole header of the FASTA file. So this is our first sequence that we imported in the Mega. How to import the second file? Go to edit button, insert sequence from file. In this case, I have this 4.fasta, which is for another crow species. So you can see the first sequence that I have is Corvus manidolius, and then second is Cobrae. So these are the different species name. Genus is Corvus, that means crow family. Okay, so this is how protein sequences look like in the Mega. If you will scroll this tab further down, you will see till the end the sequences are there. In different color, it's for the different amino acids, and for example, the letter are like this is first amino acid, methionine, isoleucine, and each letter is encode for a amino acid. Sometimes we don't, sometimes for all amino acid, it's not necessary to know what amino acid it is. So you can maybe search, for example, let's say N amino acid, one letter code. Then it will give you that this is for the asparagine amino acid. So this is how, if for some amino acid you are not sure, you can do the Google. Okay, after that we just imported the sequence. How to align these sequences? So there are two tools in the Mega. One is this W, which is known as cluster W, and there is another which is muscles. That means these are the two algorithms or programs that align these sequences. They are inbuilt in this software. So let's say I'm using muscle and align protein. Then it will ask that select all. So okay. And for this, you don't have to change these settings. These are by default settings for the alignments. Like these are the different matrices that is going to use the align these sequences. So click OK. And that is how your sequence got aligned. Okay. So this is, we know that this is ALX1 gene, and we are looking for two species from the phylogeny, from this phylogeny, and you can see around 6 million years ago, they shared a common ancestor. That means that is how they arrive from one common node, and that's why we are saying these are the sister species. So when this, the at the phylogeny level, if something are very similar, or if they are, we are calling that they are arrived from a common ancestor. This is how at the sequence level you can see wherever the star is, the amino acid is same. That means there is no variation at sequence level for this gene. Yeah, it's almost like conserved. Okay, what will happen if we will take maybe further species? Maybe let's say this one. Go to NCBI here and then download protein, one sequence per file. And then go to edit, insert sequence B5, and then align. Okay, so we found there is one, oops. Let's go from starting. Okay, so site number 53. That means for this ALX gene, which is highly conserved in the vertebrates and in birds, which is responsible for the beak shape, you can see that when we added a very diverse species, the changes occurred at the sequence level. That means this Q, that means this glutamine amino acid is only present in the fasciata but absent in the crow family. At the protein level, whenever you will see this dash, that means this is the gap, and this gap is actually represent the absence of that sequence. That means this additional Q is only present in the fasciata, not present in the crow family. Next is there is no star at this position. That at 78th position in ALX1 gene at protein level, there is serine present in the fasciata, but the asparagine amino acid was present for both the crow family. So that is what we are saying variation. And sometimes variation does not mean that you will see lot of changes. No, if you will compare different gene, because remember we are comparing same gene, because we are only interested to see the beak shape or sizes. For that, we will consider same gene across different parts. That's why we will see only certain changes, not heavily like change, because this gene is highly conserved and we are not expecting to see lot of variation. Next change, you can see that at position 98, there was a proline in this fasciata, but serine present in both the crow family. So that is how your task for this week would be to choose species either based on this phylogeny, that all the available songbirds, or either you can do one thing, that look for maybe 20 birds if you are interested in their beak sizes or shapes, and then look onto the NCBI if their gene information are available for this ALX1, HMGA2, or some other genes that you found from the paper is related to the beak morphology. And then in next session, whatever for 20 species you downloaded, I will show that how I choose the 20 species for my interest and what I did and what I found. So I wanted to see how you will do on your data set and what you will expect to get something out of that.
Can we
Any
present
question?
it? Do we
Yeah.
have to present it in the form of a document, or do we have to put it in the Meta app and then Mega app and then show you?
Okay, so for this time being, you don't have to show me your results like right now, but later on when you will prepare your results, you need to provide the screenshots of your alignment. For example, if you are saying that at 75th position in maybe 5 birds, there is only proline amino acid, and there are in other 15 birds, there is another amino acid. So maybe you can take your cursor and just take the screenshot. So I will be see that at this position, you really found a variation that is responsible for that different beak morphology in your data set.
Okay.
Mhm. Any question, confusion regarding your task?
Miss, once like how Sir sends us the recording. So, Miss, like that, can you like just once write it down, like what we have to do so that we remember, like even after like a couple of days?
Okay, so that means I need to make a slide.
Just
Okay.
a short, concise one, just telling us, what
Mhm.
outline of what we were supposed to do.
Okay, I will send in maybe text form, like
Yeah, yeah.
step by step, like what you have to do. So you can do that.
Yes, miss. Thank you so much.
Yeah, no problem. Any other question? Okay, I guess then.
So I'll share the recordings with you tomorrow, right? And meanwhile, if somebody has questions, doubts, feel free to reach out to me. I'll connect you with Vinita, right?
Yeah, and with recording, I will also share one maybe short paragraph that what you have to do exactly. So you can follow that.
Yes, thank you.
Okay, good night, everyone. Bye. Thanks.
Bye. Thank
Thank
you.
you.
Thank you.
Yeah, bye
Thank
everyone.
you.